All right, thank you for letting me know, those of you who can't turn on your video. And so for everyone else, please do turn on your video and we will get started for today. So as someone mentioned or in the chat, we are going to learn about DNA mutations today, but to understand why mutations are bad for organisms and bad for us because we are organisms, we have to first understand how it contributes to our existence and how DNA is actually going to affect the cell and the organism, if you're talking about a multicellular organism, and just how is the information that's encoded and stored in DNA going to be expressed. And that's going to be the process of gene expression, which is our topic for today. Is it possible mutations can be good in the long term? So yeah, mutations, I think normally when we think about them, they are in a negative light because we think about how mutations can mess things up and lead to diseases such as cancer. But mutation can definitely also be a good thing. Mutations in general, just the term is referring to just changes in your DNA sequence, right? And so when we talk about evolution and population genetics next week, we'll be talking about how some types of mutations that are actually going to be good for the organism and improve the organism's fitness and their ability to survive and reproduce are actually going to be passed down because of the fact that they provide the organism with beneficial traits rather than negative traits. So yeah, we'll talk about all of those in the coming week, as well as some of those things today. And again, feel free to send me your questions or unmute during today's lecture. And because of feedback from students and parents, I decided to add in some extra practice questions in the middle. We will be having Kahoot again today at the end. So a lot of those practice questions will be directly related to things you'll see on the Kahoot. So I really recommend trying them out and just seeing if you have the right ideas in mind. So before we get started with gene expression, I actually wanted to talk about a very similar process, which is DNA replication. And to talk about DNA replication and also just to talk about gene expression in general, it's important to understand the structure of DNA and basically what is DNA actually. And we talked about it a little bit when we talked about the different types of macromolecules. And so we know that DNA and RNA are both examples of nucleic acids, right? And so both of these types of nucleic acids are responsible for carrying genetic information. And these nucleic acids are made up of nucleotide monomers. So their building blocks are nucleotides, and they are made up of chains of nucleotides. And so the 5 different types of nucleotides that we're going to talk about are adenine, guanine, cytosine, thymine, and uracil. And so these 5 nucleotides are going to be they all share this common structure as shown in the upper right here, where you have a nitrogenous base, a sugar, and a phosphate. And so the sugar and phosphate group will form the sugar-phosphate backbone of DNA, which some of you might have heard before. And then the nitrogenous base, which is going to be made up of one of these structures here, is going to determine the identity of your nucleotide and also interact with each other to form the complementary pairs in your DNA helix, which we'll see on the next few slides. And so one thing to know about nucleotides is that not all of them are present in both DNA and RNA. So adenine, guanine, and cytosine are going to be ones that you see in both DNA and RNA. So you have DNA versions of the adenine, guanine, cytosine nucleotides, but you only have and you also have an RNA version of each of these 3. But for thymine, you only have a DNA version of the nucleotide. And for uracil, you only have an RNA version of the nucleotide. And this is going to make things a little bit more tricky when we talk about DNA replication and gene expression, but hopefully it's not too complicated. And so remember that the sugar molecule in the sugar phosphate backbone is also going to be different between DNA and RNA. And so that's why even though these the A, G, and C nucleotides are present in both DNA and RNA, we're not talking about the exact same molecule, although it might be easier to just think of them as in terms of the identity, whether it's A, G, C, T, or U, rather than thinking about the molecular level, just for now. And so with DNA and RNA, they are both going to be in the form of polynucleotide chains, which are, again, chains of nucleotides that are linked together by covalent bonds. And this is something that we mentioned really briefly during our previous lecture, but now we have to kind of actually understand why this is, which is, and that thing is that these polynucleotide chains, these DNA sequences and RNA sequences have something called directionality. And what that means is that with a given chain or a given sequence of DNA, you're going to have two chemically distinct ends. And those distinct ends are called 5' end and 3' end in DNA and RNA. And this is really important because new nucleotides are only added to the 3' end of a growing chain of a growing polynucleotide chain. And so if you mess up the different ends, that's going to really gravely affect what your DNA result is. And as you guys will see when we talk about gene expression, it's going to affect what your final product is going to be. And so it's really important to keep your two ends straight and to kind of keep track of where they are relative to each new DNA or RNA sequence that you're generating. And so why is it called 5' and 3' ends? That seems kind of arbitrary. And so the reason goes back to the molecular structure of the nucleotides. So if we go back to this previous slide, we said that all nucleotides have this sugar molecule in its in the structure of the nucleotides. And with this sugar molecule, so the one shown here is going to be a ribose, so with RNA, but same thing with DNA, where the carbons in this structure are actually going to be numbered. And the way that they're numbered is 1', 2', 3', 4', and 5'. And so this is where the name for the ends come from. The 3' carbon, or the 3' end, is going to be the end that corresponds to right here, where the new nucleotides are being added. And the 5' end, which is where the phosphate group is going to be connected, is going to be the end that is either at the very beginning of your polynucleotide chain or the end that gets attached to a growing chain. So you can think of this as if you're trying to like thread beads onto a string, except the beads are not circular and instead they have a asymmetrical shape. So let's say that these are your beads, okay? And we'll say that this is a 5' end and this is the 3' end. So with DNA, the 5' end, if this is your first nucleotide, the 5' end is not really going to interact with anything. And rather it's going to form a bond with another nucleotide the 3' end of one nucleotide is going to form a bond with the 5' end of another nucleotide, and then so on and so forth as you build your chain. But once your chain kind of gets started and you have 2+ nucleotides, then your new nucleotides can only be added onto the 3' end, as we mentioned before. And so because of this, once you finish your entire DNA sequence or RNA sequence, in this case of 3 nucleotides, you're going to have one end that has the 5' phosphate group that's not in a bond, so we call that exposed. And then on the other end, you're going to have the OH group not involved in a bond and exposed. And so notice that if you were to look at a nucleotide that was in the middle of the chain, or if you were to switch the ends up, that wouldn't work anymore because the phosphate group here is involved in this covalent bond, and then the OH group is involved in this covalent bond. And so that's what gives this directionality to the DNA and RNA sequences. And this is going to be important for later. And so you don't really need to understand too much about the molecular basis for why. It's just, this is something that people always have questions on, which is why we are talking about it. But for the most part, I think the most important thing to remember for honor biology is that DNA has a 5' end and a 3' end, and that these ends are different from each other. So you have to know which end of the DNA you're looking at and which direction you're reading your DNA script. And we'll see some examples of that in a little bit. And so since we're going to start with DNA replication, we're going to start by talking about DNA structure. So forget everything before was both DNA and RNA, and now we're just going to focus on DNA structure. So DNA, as you guys probably already know, is double-stranded in our cells. And so you have two strands of DNA that are complementary to each other, and together they form what is called a right-handed helix. And the right-handed helix, I can't remember if we talked about this last time, but what it means is that if you take your right hand and you kind of take your thumb and you curve your fingers in, the direction that your fingers are curving in is going to be the direction that the helix moves in, looking at it from the top. So you guys can try that on your own. I think the video might be mirrored, so it might be a little confusing, but if you turn your hand, your right hand is going to turn in the opposite direction to your left hand. And that's why there's a difference between a right-handed helix and a left-handed helix. Something else to know about DNA structure is that the two strands of DNA in the helix are going to run in opposite directions, and we call this anti-parallel. And so what does it mean that they run in opposite directions? It doesn't mean that they're actually moving past each other, but rather one strand of DNA is going to go from the 3' end on the top to the 5' end on the bottom. This one is curved, so it's a little harder to follow. But if you follow the strand down, you can see that it starts on the 3' end and then ends on the 5' end. And then the other strand will be the opposite, where the top is going to be the 5' end and the bottom will be the 3' end. And that's what it means to be anti-parallel strands. And so these two anti-parallel strands are involved, are both interacting with each other to form this double helix. And when they interact, this is dependent on their nitrogenous bases in their nucleotides. So since we're looking at DNA, we're only looking at A, T, G, and C. So no uracil, no U for now. And we have what are called complementary base pairs. So the nitrogenous bases, the nucleotides, are going to be matched up with each other such that A, if you have A on one strand, so seen in the red here, you're always going to have T on the other strand, seen in the orange here. And then guanine and cytosine will be a pair. So if you see blue, you will see green on the other strand. And then vice versa. If you see green, you'll see blue on the other strand. And so this is really important because this is the foundation for how DNA replication and also gene expression occurs. Because when you make new DNA strands out of old template DNA, you're adding in nucleotides and you're adding in complementary nucleotides to be specific. And that's how you ensure that the new DNA you create is an exact replica of the DNA that you started out with. Because otherwise it wouldn't be DNA replication, you would just be making DNA randomly, and that's pretty bad for a variety of reasons. And so it's really important to remember these complementary base pairs. And something else to mention is that with RNA, instead of AT pairs, you're going to have AU pairs instead, because of the fact that you don't have thymine RNA nucleotides, you only have uracil RNA nucleotides. And so the GC pairs stay the same, but instead you will see A and U rather than A and T. Okay, so before we dive into DNA replication, are there any questions about what we just talked about? So DNA structure, complementary base pairs, etc. No? Okay, cool. Again, let me know if a question does come up. And so DNA replication, as I already mentioned, is the process by which you create an identical copy of your DNA from a template DNA strand. And this would be a double-stranded DNA helix. And so this process is semi-conservative by definition because you start off with the original template shown here in the blue. So that's going to be your original DNA helix. And what happens is that both strands in your original helix are going to act as templates for your newly synthesized DNA during DNA replication. And so the newly synthesized DNA, or the new nucleotides that are added, are shown in the red here. And so you can see that your final products contain both a template DNA strand and a new DNA strand. And so it's semi-conservative because you keep, each of these keep half of each is original DNA. And so this is actually the process where most DNA mutations occur. And we'll talk about some of these mutations, and as I mentioned towards the beginning of class, and we'll see kind of how they can affect gene expression as well. Because when you replicate your DNA, you're replicating your DNA during things like S phase, where you're preparing for cell division. And so if you replicate your DNA and something goes wrong and you have a mutation, that's going to change or potentially change the products that you're going to get in one or both of your daughter cells if we're thinking about mitosis. So DNA replication, we're not really going to go into too much detail, but there are some pretty key players that I put in bold. And we didn't really get a chance to talk about enzymes because this course is pretty short. We only have 10 days, but the helicase, topoisomerase, primase, all of these things that end in ase are going to be enzymes. So they are special proteins or a class of proteins that are going to help speed up different reactions. And in this case, you can think of them as having specialized functions that speed up the process of DNA replication overall. Okay, going back really quick, question in the chat. So this diagram here, it's showing DNA replication, although the diagram is a little bit weird without understanding what DNA replication is. And so what happens is when you have your original DNA template, it's actually going to get unwound by a special enzyme called helicase. And when it gets unwound, I think that's the past tense for it, what happens is that both of these blue original strands are going to act as your template strands. And so another enzyme is going to come in and add in new nucleotides based on the information in your template. And so that's shown in the red here. And I think it'll make a little more sense once we go through the different steps of DNA replication, but let me know if you are still a little bit confused towards the end. So the first step of DNA replication is where helicase unwinds the DNA helix. And so when your DNA is in the helix form, let's go back a few slides. Let's see. So when it's in the helix form, you can see that your complementary base pairs are actually going to be interacting with each other. And something that I forgot to mention is that this interaction is dependent on hydrogen bonding. So there's hydrogen bonding between the A and T pairs, and then there's hydrogen bonding between the C and G pairs. And so normally your DNA doesn't just kind of come apart. So instead we need an enzyme like helicase or some other molecule. So in this case, it's helicase to unwind the DNA and separate the two strands so that we can actually access them and add new nucleotides to them. So if they're attached to each other, you can think of them as being kind of glued together. If your two strands, if the nucleotides are glued together, there's no way for you to add in new nucleotides to each side, right? So that's why we need helicase to split your DNA helix and to create those, to expose those two template strands for us to use. And something that works together, kind of side by side with helicase, is called topoisomerase. And this is another enzyme. And what it does is it works kind of behind the helicase, closer to, it's on the other side of the helix that's not getting unwound. And so what it does is that it prevents DNA from getting too tightly bound as it's being separated. So I'm not sure how many of you guys have experience with like just messing with yarn or string or anything similar to that. When you try to split a string into smaller pieces, so like if you take a piece of yarn and you try to pull it apart, because they are wrapped together, when you pull it apart, it actually can get tangled towards the end and then you can't unwind it any further. And so the topoisomerase basically helps prevent that from happening when you unwind the DNA helix, because it just helps turn the string, or in this case, your DNA helix, to release the pressure of the helicase unwinding it from one end. And I'll show you guys a video in a little bit that will hopefully kind of provide an image for what this is, what this process is doing. And so after your helicase separates the DNA helix and reveals, one second, so why would it get too tightly wound if the helicase is unwinding the DNA helix? Okay, so let me find something, let's see. All right, so kind of poor, not the best modeling, but these are two chargers, but think of them as your two anti-parallel DNA strands. And so if they are wrapped around each other in a helix form, this is not quite right, but you guys get the idea. So if they're wrapped together, and what happens is your helicase is unwinding it from one end. And so if you just unwind and unwind and unwind, it actually is going to kind of build up pressure, and you can see how it gets more kind of stuck together towards like where it's still stuck together as you're trying to pull it apart. And so that's kind of what topoisomerase prevents from happening, because if this happened, then it would be really difficult to actually separate your two strands of DNA. Does that kind of make sense? Yeah, a lot of these things, I think it's makes it a lot easier if you can associate them from phenomenon that we see in everyday life. And so it's, it's not quite exactly what happens, but it's a good enough metaphor / analogy. And so the next step after you've exposed the template strands is another enzyme called primase is going to come in and create RNA primers. And these RNA primers are short sequences of RNA nucleotides that are built from the template DNA. And these primers are going to be complementary to the template DNA. So wherever you see an A in the template DNA, you're going to add a U nucleotide because it's the AU pair. And then wherever you see a C, you're going to add a G RNA nucleotide, and so on and so forth. And it seems kind of weird for you guys to, or for us to add RNA primers when we are trying to replicate DNA. But what this primer does is that it serves as a starting point for our next really important enzyme called DNA polymerase. Without the primers, without the RNA primers, DNA polymerase would not be able to bind to your DNA and to your template DNA and start adding new nucleotides, which is the next step. And so it's really important that we have these RNA primers being added to the template DNA so that the DNA polymerase enzyme can come in and bind and then start the next step of the process. Question? Yeah, so primers are going to be short sequences, in this case, of RNA nucleotides. And so, DNA and RNA is just really long chains of nucleotides. And so a primer is just it's a special name for a short sequence of nucleotides, in this case, RNA nucleotides. And the exact sequence is not it doesn't have to be the same. It just depends on which part or what the template DNA is. And the reason why it's called a primer is, if you think about it, like priming something, like if you're priming, I don't remember, I think it's like the engine or something like that, you're getting it ready to go. And so that's kind of why it's called a primer, because it gets the DNA polymerase ready to go and start the next step of the process. And so after your primase has added the RNA primers, your DNA polymerase can now come in and bind and synthesize the new strand and the new DNA by adding complementary DNA bases to the template DNA. So before, when we were making the primers, we were adding complementary RNA bases, and now we're actually adding complementary DNA bases. And so that's kind of shown here, where here's your parental molecule of DNA or parental strand of DNA, both shown in blue. And so after it gets separated, and the primer is not shown here, but what the DNA polymerase does is that it reads the nucleotide, it reads the base that is on the parent strand, and then it adds the complementary DNA base for that. And so it reads A and it will add T, and then reads C, adds G, reads G, adds C, et cetera, et cetera. And so by doing this, what you can do is create two identical DNA sequences from your original strand of DNA. And so if you kind of compare these two, you can see that originally this was A and T. And so because you're adding in the complementary base pairs, you have A and T, C and G, G and C, et cetera, again. So this strand is going to be identical to the parent. And then this, even though you're adding in different nucleotides from here, but because they're complementary, you have A and T, C and G, A and C. Sorry, that's not A. It should be G and C, A and T. That's also going to be the same as the parent DNA. And so by doing it this way, you can ensure that your DNA replication products are going to be identical to each other and to the parental DNA. And something that is really important about DNA polymerase is that it only adds nucleotides in the 5' to 3' direction. And this is related to the fact that you add new nucleotides to the 3' end of your growing nucleotide chain. And so so RNA polymerase can also only add nucleotides in the 5' to 3' direction. And so this is not super important for DNA replication, but it's going to be very important for gene expression because that's going to be a little more tricky. And so during the process where DNA polymerase is adding in new nucleotides to create the new DNA strands, there's actually going to be a slight difference in how the two strands are being created because you have two template strands. And one of these strands is going to be called the leading strand, while the other is going to be called the lagging strand. And there's a difference in how they're made because of the fact that DNA polymerase only adds nucleotides in the 5' to 3' direction. So both of these, right here, the gray is showing you the original template DNA, and the fact that it kind of closes in like this is because the helicase is actually still working. This helicase is still working to unwind the rest of the DNA, but DNA polymerase has already started adding nucleotides. And so this creates a little bit of a problem for this lagging strand here. Because with the leading strand, you create your primer in the orange. Let's say that this is the end of your DNA. So you create your primer in orange, and your DNA polymerase, which is this blob here, can add in new nucleotides in blue. And as helicase unwinds the DNA, this DNA polymerase can just keep going. And so as it unwinds, DNA polymerase can just add more. And they can do that because they're synthesizing it from the 5' to 3' direction, which just happens to be pointing in the direction of the unwound DNA. With the lagging strand, however, because the two strands because your DNA helix is, the two strands are anti-parallel. So if one of them runs from 3' to 5' in this case, the other template must run from 5' to 3'. And so if it's running from 5' to 3', then your DNA polymerase has to move away from where the helicase is, away from what is called the replication fork, which is where this where the DNA helix is getting kind of opened up. And so because it's moving away, as indicated by the arrows here, every time that the helicase exposes more template DNA, a new primer has to be added closer to the replication fork so that the DNA polymerase can bind and then synthesize this segment of DNA. Because if you think about it, like at an earlier point in time, it could be that this area was the only part of DNA that was exposed. And so once your DNA polymerase kind of binds and then creates this section, the helicase has exposed more DNA. And so now it has to unbind, and then another primer has to be laid down, and it has to go back and then start again from here and then synthesize until the previous primer. And so because of this, the lagging strand is synthesized in pieces rather than in a continuous strand, such as with the leading strand. And what these fragments are called are Okazaki fragments. And so obviously it won't do you any good if your DNA is just in random strands, random fragments. And so after everything is synthesized, so after the entire DNA sequence or original helix has been unwound and all of the new nucleotides have been added, the primers will actually be taken out and replaced by DNA nucleotides, and then another enzyme called Helicase will come back and basically connect all of those gaps in between the fragments so that it forms a continuous strand. And so the video on the next slide should show you guys what that looks like in an animation, which is pretty nice. And so I think the really important thing to understand for DNA replication is why is there a difference between the leading strand and the lagging strand? And so if you guys have questions, please let me know, because this does get pretty confusing. And the reason why the DNA polymerase can only move like this is because, again, it has to synthesize from 5' to 3', which means if your DNA strand is running from 5' to 3', then it has to run in the opposite direction to that, if that makes sense. Okay, and so this is a 3D animation of DNA replication, and it's a good review of everything we've discussed so far. And please let me know if you guys can't hear the sound, because I think I shared sound, but I'm not sure.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called Helicase and results.
Sorry, I meant to pause it for a second. One second as I pull it up.
Wherever there's how.
Okay, so what I wanted to point out was that with the helicase, so the topoisomerase that we talked about, which is not shown in this video, would be somewhere on this side of the DNA helix, so that it's kind of turning the DNA a little bit as it's unwinding the helix. And it looks a lot simpler here because they're showing the DNA helix as two parallel strands where it's kind of straightened out. So if you imagine that this is all twisted like in a helix, it would kind of make sense why you need topoisomerase there to prevent it from getting tangled in a sense.
Done by an enzyme called Helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA Polymerase binds to the primer and will make the new strand of DNA. DNA Polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay. So hopefully that is beneficial to your understanding of DNA replication. And so here is a practice problem. Let's take around 3 minutes to work on this, and so we'll come back at 38, and I would like to get a volunteer to share their answer. What tells the DNA to stop replicating? So that's a pretty good question. I think if I remember correctly, I think it just replicates until the original DNA template is done. But I will double check on that. I think it will replicate the entire strand of template DNA that you started out with, and that's when it stops replicating. But I'll double check on that. And so one thing to remember about DNA synthesis is that because they are synthesizing a new DNA strand. And remember that those two strands just kind of join together to form the double helix again towards the end. So they also have to be anti-parallel to each other, which means that your newly synthesized strand is going to be anti-parallel to your template strand. And so think about how that would affect the 3' and 5' ends in your complementary DNA sequence. Yeah, that's a great question. So is there a reason why it is separated into groups of 3? Yes, there is, but it's not relevant for DNA replication. We will be talking about it when we talk about gene expression. So the groups of 3s, or they are grouped into 3s because that's how the RNA is going to be read during gene expression and be converted into protein. And so I just put all of them in the 3x3x3 kind of configuration. Yeah, and we're actually going to be using this same sequence for all of the practice questions today. And so you can kind of, if you write down your answer on a sheet of paper, then you can kind of see like what the difference would be if you were doing DNA synthesis with this template versus gene expression or transcription, to be specific. All right, it is 38. Would anyone like to volunteer to share their answer? Just raise your hand in the Zoom, I think, or let me know in the chat if you would like to volunteer. Anyone? Okay, great. Andrew, go for it.
Okay, here's what I have. So first we start off with the 3', and then we go ATG, TGC, GGC, and AGT, and then we end with 5'.
Yeah, that is really close, actually. Good job on getting all of the bases. And so the only thing to change is the 5' and 3' ends. And so, like I was trying to hint at earlier, when you make the new strand, it's going to be anti-parallel to the template strand. And so when it starts from the 3' end here, that means your newly synthesized sequence is going to start from the 5'. But really good, very, very, very good, good job. Okay, so here is the answer. So just like Andrew said, you just find the complementary pairs for your template DNA. Okay, any questions on this question? All right, cool. So now we can talk about gene expression, and it's actually 39, so we can take a break now just to kind of give your brains a rest before we dive into more materials. So let's come back at 45 or 44, and we will continue then. Let me know if you have any questions in the meantime. Okay, cool. So my goal for today is to actually end like 10 to 15 minutes early so that everyone can participate in the Kahoot. Yeah, hopefully we'll get through everything, but we'll see. And so now we can talk about gene expression, which is how DNA is actually going to be converted into more visible and observable traits. And so we already talked about genes a little bit when we talked about mitosis yesterday. And we know that genes are found on chromosomes and that they are kind of the functional unit of a chromosome. And they are also considered like the kind of a very small unit of genetic information. And so, okay. And so the way that DNA is actually going to be converted into products or into things that are going to affect the cell and affect the organism as a whole is by creating gene products. And so the information encoded in the DNA sequence is going to be the instructions for making these gene products. And these gene products can be either proteins or RNA. And we're actually going to focus on the making of proteins as a gene product. Although RNA is also very important to a variety of things, but we're going to focus on protein synthesis mostly. And so gene expression is just how DNA can determine an individual's traits. And because we're going to focus on the protein synthesis aspect of gene expression, this is a two-step process that involves something called transcription and something called translation. And these are the two steps represented by the arrows here, where transcription is the making of RNA from DNA, and then translation is the making of protein from RNA. And so you can't just make protein from DNA, and that's why it's a two-step process to go from DNA to RNA, then to protein. And so we're going to start with transcription first, and we're making RNA from a DNA template. And this is going to be pretty familiar sounding because it's, if you look at it very broadly, it's very similar to the DNA synthesis process that we just talked about, because you're still making a copy of DNA, except you're making this copy using RNA nucleotides, and you're only making this RNA sequence based on one strand of the DNA rather than both strands of the DNA. And so we'll talk about which strand of DNA we're actually using, and that's also going to be related to the 5' and 3' ends of your DNA directions. And so the transcription process is catalyzed by an enzyme called Rna Polymerase. And so don't get this confused with DNA polymerase, which is the enzyme we saw in the DNA synthesis and DNA replication process. So RNA polymerase is very similar to DNA polymerase, except it adds RNA nucleotides instead of DNA nucleotides. And so just as DNA polymerase, RNA polymerase is going to also add new nucleotides to the 3' end of the growing chain. So it synthesizes your new RNA script from the 5' to the 3' direction. And since we're adding RNA nucleotides, you're only going to see the bases A, U, C, and G. So no T in this case. And so what happens, how does this really work with converting DNA to RNA? And so that's shown in this diagram here, where your template DNA, shown here from 3' to 5', is going to be acting as a template, as the name suggests, for this RNA that's being synthesized. And so the RNA polymerase is going to read the template DNA just as before, and it's going to add the complementary RNA bases. And so when it sees an A, instead of adding a T, it's going to add a U, because that's the complementary RNA pair for adenine. And then where it sees a C, it'll still add a G. And then vice versa, where it sees a G, it'll add a C. And then where it sees a T, it will add an A. So this pair actually stays the same, but only because A is present in RNA, but T is not present. So if you see a T in the DNA, you'll have an A in the RNA. But if you see an A in the DNA, you're going to see a U in the RNA. And so just make sure you guys don't get that part confused. And transcription can be broken down into 3 stages: initiation, elongation, and termination. And these are just really very descriptive names, because they basically tell you what happens during each step. Initiation is when the transcription process begins. Elongation is when your RNA sequence elongates or you get more nucleotides added. And then termination is when it ends. So really easy to remember. And so initiation, how does it start exactly? So RNA polymerase has to bind to the DNA. And rather than having RNA primers, RNA polymerase is going to bind to the DNA in a region called the promoter region. And so the promoter region is a sequence of DNA within your within your original DNA that is going to direct the binding of RNA polymerase. And so it'll know to bind here, and this is where it will begin the transcription process. And so here's kind of where the transcription process differs from DNA replication, because you're not using a primase to add RNA primers, and you're also not using a helicase to split your original DNA helix. Instead, the RNA polymerase is going to work together with some other molecules to split the DNA and create what is called a transcription bubble. And so this transcription bubble, as you can see, will expose the 2 strands of DNA so that new nucleotides, RNA nucleotides, can be added by the RNA polymerase. And so, as I mentioned before, with transcription, you're only making 1 RNA strand. And so a single RNA strand, which means you're only going to use one of the DNA strands as your template, and that's going to be your template strand. So when we talk about template strand in this context, it's different from when we called from what we refer to as the template DNA strands in DNA replication, because in that case, both of the strands of DNA are going to, of original DNA are going to be the template strands because they act as templates. But in the case of transcription, only one of them is going to be the template strand, and then the other is going to be called the coding strand. And so RNA nucleotides are only going to be added by RNA polymerase based on the template strand. So they're complementary to the template strand. And you can see that, again, the 3' and 5' is flipped in the 2 strands because they're still going to be anti-parallel to each other. And the coding strand, even though it's not getting any RNA nucleotides added, it's still pretty important and can provide you with a lot of information because it's nearly identical to this RNA strand that's being made. And it makes sense if you think about it because both of these are complementary to the same template strand, right? So other than the fact that you have RNA nucleotides here and DNA nucleotides here, it makes sense that the bases would be almost exactly the same, except for all the T's would be used in this case. And so one thing to be careful of when you're looking at questions like these is that sometimes you're going to be given the template strand DNA sequence and asked to find the RNA sequence. And in that case, you should just find the complement. But if you were given the coding strand sequence and asked to find the RNA sequence, you would have to make sure that they're nearly identical and just change all the T's into U's. And notice that the 5' and 3' ends actually match up between these two. And that's because they're not complementary to each other. They're both complementary to the same template. And that's why the 5' ends and 3' ends are even going to match in that case. And so during elongation, RNA polymerase is going to add nucleotides from the 5' to 3' direction. And so it's going to add in more nucleotides to this side and just move along the template strand. And, as all things, transcription also must end somewhere. And so termination of transcription is going to be when your RNA transcript is released from the template DNA. And one thing to note about RNA is that it's actually going to be single-stranded for the most part, especially in our cells. So when you have the synthesis of this RNA, as you'll notice if you think about this diagram here, you're only going to have a single strand of RNA. It's not going to be in a double-stranded format, and it's not going to be in a helix. And so how does transcription end? And so just as how we had a promoter sequence to tell us kind of where to begin with transcription, we also have a terminator sequence that tells us where to end. And so this is a sequence of DNA that just tells the RNA polymerase to stop transcription and to release the RNA transcript that it's made. And so once the RNA transcript is released, it'll actually go through some modifications before it becomes what is called mRNA or messenger RNA. And so messenger RNA is the kind of the final product of what will move on to translation and be converted or be decoded into a protein. But so the RNA sequence or the RNA strand that you made during elongation is not quite ready to move on to that step yet. And so it has to go through these modifications. And so with all RNA, what you're going to do is you're going to add something called a 5' cap and a 3' poly A tail to the RNA sequence. And so RNA sequence is represented in the red pinkish color here. And then the 5' cap is a guanine nucleotide. And then the poly A tail is a string of adenine bases. And so those are added to all of your RNA sequences that are made during transcription. And then you're also going to have splicing for the most part. And this is removing certain parts of this pre-mRNA, just the RNA that it's not quite ready to move on to the next step yet. It's called pre-mRNA. And so when you remove certain sections, you're actually removing pieces called introns, and then you're keeping pieces called exons. And so this is kind of what it looks like. Your pre-mRNA, so immature mRNA, is going to be made up of a varying combination of exons and introns. And this is actually going to be determined kind of by evolution. So we're not going to talk too much about how do we know if something's an intron, how do we know if something's an exon, just know that they exist in the RNA. And so what happens is that you have these special enzymes that can just splice the DNA and essentially kick the introns out of the sequence. And then it gets ligated back together again. And so now you have an mRNA that's ready to move on to the next step of translation. And it only contains the exons you want. And the reason why this step matters is because your DNA is going to, or your DNA sequence is going to affect the protein product that you produce. And so if you have these introns, you're going to have additional nucleotides in your sequence that are not necessary for your protein product. And so I think it'll make more sense again once we talk about translation. But if you had extra nucleotides, you could end up with extra amino acids, and that could throw your entire protein structure out of whack. And so this is why your mRNA has to go through these modifications. And so once you've generated the mature mRNA and it's ready to move on to create protein product, then you're going to undergo something called translation. So the second step of gene expression. And in translation, you're going to build proteins from the information that's encoded in the mRNA. And something that is really helpful to translation and questions about translation is this codon chart right here. And so this codon chart is going to tell you how to decode mRNA into a protein. And so we'll see some examples of that in a little bit. And so this is where that earlier question comes into play. So someone asked earlier, why was the DNA sequence split into groups of 3 nucleotides each? And that's because of what are called codons. So the mRNA nucleotides in your mRNA sequence are read in groups of 3 called codons, and they are read from 5' to 3'. And this is also important because if you're reading something from 5' to 3', so reading something, for example, from left to right, that's going to be different from reading that same thing from right to left, right? And so in the case of your codons, if you have CUU, right, cytosine, uracil, uracil, if that was 5' to 3', it would encode for the amino acid LEU. But if you were to read it from 3' to 5', where you have UUC, that's going to be PHE, which is a completely different amino acid. And that's going to affect your final protein product. And so that's why it's important to remember that your mRNA is read from 5' to 3'. And so each of these codons are going to code for a specific amino acid. However, because as you guys are probably all mathy people, you have 3 nucleotides and 4 possibilities for each of these nucleotides, right? And so you have 4 to the 3rd possible, what's it called, combinations of these letters. So 4 to the 3rd possible codons that you can have. And with amino acids, if you guys remember way back to our discussion of proteins, you only have 20 common amino acids that you see in our proteins. And so you have 64 different codons possible and only 20 amino acids. And so some of them are going to encode for the same amino acid. And this is going to matter when we talk about the mutations and how the effect of different mutations. And so, for example, all of these here encode for the same amino acid. Both of these encode for the same amino acid. Both of these encode for the same amino acid, etc. And the kind of the exception to this, to the fact that codons encode for amino acids are the stop codons. And the stop codons are signals that tell your transcription process to stop, and that signals the end of your amino acid sequence that you're generating. And so this is also going to be pretty important, but these are not amino acids. Question in the chat. What's a polypeptide? So a polypeptide chain is just a chain of amino acids. And I'll do a quick review of protein structure on the next few slides. So you'll see again then. And something else to remember is this codon right here, AUG, which is highlighted in red. This is also known as the start codon. So it's usually at the beginning of your polypeptide sequences, of your amino acid sequences. And this is also an amino acid at the same time. And so it's a little different from the stop codons, but this is usually found at the beginning. And if you were asked, like, what is the start codon? It would be AUG. And so translation is going to occur in the ribosomes, which we talked about during the cell structure lecture. They are structures within your cell that are responsible for protein synthesis. And so they can either exist just floating around in the cytoplasm, or they can be embedded in the ER. And so they are going to help, or they're going to be primarily responsible for turning mRNA into amino acids, or not turning, translating. Yes. And so a quick review of protein structure. Still a little confused on how to read the chart. Okay, yeah, so let me go back really quick. Sorry, I forgot to explain that part. And so this chart, it's kind of like Battleship. So when you are given a codon, so let's say we're looking at AUG, all right, just to make it easier. AUG, your first letter, you're going to find it here. And so A, and then you know that it's going to be within this row here. And then second letter is U, and so you know that it's in this box. And then third letter is G, and so you know that it's this last one. And so whatever your codon is, you can just kind of search it up like this to find where it is in this chart. And then it'll tell you which amino acid is encoded by that codon. And so everything in the 3-letter kind of sequences here are going to be the codons. And then these abbreviations here are going to be the names for the amino acid that they encode. Does that make sense? Okay, great. Yeah, sorry, I forgot to talk about that part. And so with protein structure, because we're making proteins, it's kind of important to know or to remember, what is their structure. And so proteins, as we know, are made up of amino acids, and protein structure can be divided into 4 levels, as we talked about. The first level is going to be the unique amino acid sequence in the polypeptide chain. Sorry, it's kind of cut off by the picture here, but that's going to be the kind of the level of structure that's determined by gene expression. This is going to affect secondary structure, tertiary structure, and sometimes quaternary structure, but it's going to affect it indirectly. So if you think about secondary structure and tertiary structure, so secondary structure is local folding of the polypeptide chain, while tertiary structure is the 3D folding of the polypeptide chain as a whole. And then quaternary structure is if you had multiple polypeptide chains that were folded, and then they come together and interact with each other and then kind of fold again in a certain configuration. All of these kind of levels of structure are dependent on interactions between the amino acids. And so if your amino acid sequence was different from, or was changed from whatever it was originally supposed to be, that could change your secondary structure, your tertiary structure, and your quaternary structure. And so if you guys remember, the analogy that we used when we first talked about this is that your amino acids are kind of like beads that are only magnetic for certain other beads on a string. And so if you change the sequence of those beads, then they're going to interact with each other. They're still going to interact with the same partners, but if they're further apart, it's less likely that they'll interact. And so like the interaction patterns are going to change, which is going to result in a different folding pattern for all of these other levels of protein structure. And so even though the mRNA sequence is directly only determining the amino acid sequence, it's going to indirectly affect all of these other things, which is why it's so important to have the correct DNA sequence, the correct mRNA sequence in order to create the right proteins. And because we talked about how proteins are really kind of sensitive to changes in their shape. So if you change the shape of the protein, you could potentially lose function or change its function. And so if you're changing its structure, you're going to affect its function. And that's why it's so important for this step to be kind of accurate. And so this is showing you the 20 common amino acids, and they are grouped into these 4 groups by their special kind of properties. And so the amino acids here, so you can see there's their full, they have the full names and then the 3-letter abbreviations that you saw in the codon chart. And so all of these here are going to be charged amino acids, or amino acids with charged side chains. And then these are going to be polar uncharged side chains. And these are going to be hydrophobic side chains. And these are some special cases, but you don't have to memorize this, of course, but I just wanted to show that depending on which amino acid you're talking about, they could have very drastically different properties, because if you think about like a hydrophobic molecule versus an electrically charged molecule, they're obviously going to interact much differently with their surroundings compared to each other. And so another reason why it's so important to have the correct amino acid sequence in your protein. And so how does this process occur exactly? And we're not going to go into too much detail, but this is dependent on ribosomes and the help of what are called tRNAs, or transfer RNAs. And so the ribosome is represented by this green blob here, and the ribosome has 3 sites. And so you have the A site, the P site, and the E site. And the tRNAs are these special structures that are mostly made up of RNA, but they contain what are called anticodons, and they also are kind of carrying amino acids. And so what determines, which amino acid it's carrying? That's going to be dependent on the anticodon. The anticodon is going to be a 3-nucleotide sequence that is complementary to the mRNA codons. And so if you have AUG, the anticodon to that codon would be UAC, so complementary to each other. And it doesn't really matter the 5' to 3' in this case, but it would just be flipped as well. And so with the anticodons, the anticodon that correspond to a special codon, if the tRNA contains it, then it's going to be attached to that amino acid. So if you have the anticodon for AUG, then that tRNA will be carrying MET, which is the amino acid encoded by AUG. So let's go back really quick. So let's look at the second one. The second one, the sequence is UGG, again from 5' to 3'. So UGG, not the boots. So UGG, let's see. So that's going to be TRP, tryptophan, which is one of the amino acids. And so if we go back, we can see that the anticodon, ACC, is going to be on a tRNA that's also carrying the TRP tryptophan amino acid. And so basically the tRNAs act as little shuttles that help bring the correct amino acid to the ribosome so that it can be added to the polypeptide chain. And so tRNA will arrive at the ribosome and be in the A site, and then it will basically give its amino acid to whatever polypeptide chain is growing. And so the TRP will be attached to the MET, and then the tRNA will kind of move through the different sites and then leave once it reaches the E site. And so that's kind of all you need to know. You don't really need to know exactly the molecular basis for what's happening, but it's a bunch of different interactions and binding and intermolecular things. So we don't really need to know the details of that. What's important is that the tRNAs have these anticodons that are basically going to correspond to the codons in your mRNA, and they're going to bring the correct amino acid to your ribosome to be added to your amino acid sequence. And so if you can kind of picture it, once you add this amino acid, once this tRNA arrives and brings this amino acid, another one is going to arrive for UUC, and then that's going to add the next correct amino acid to your chain until you kind of reach a stop codon and synthesize a longer polypeptide chain. And so here's a practice problem. So this is the same strand of DNA that you guys saw before, except this time we're not talking about DNA synthesis. So kind of just erase everything you just did from the last practice problem. And you're given that this is a template strand of DNA, and you need to find the amino acid sequence that would be produced by translation. And so I want you guys to actually, go through all the steps. And so I'll take a volunteer for the mRNA sequence as well. And then I'll take a volunteer for the amino acid sequence. And so just for the sake of this question, forget about like splicing and all of that. Just treat this as your mRNA, or treat this as, or your mRNA would just be whatever it's supposed to be. No modifications. Let's see, right now it's 11, so let's take, we can take 6 minutes, so until 17. And let me know if you want to check your answers with me, just in the chat, send them to me directly. Or if you have any other questions about, DNA translation or DNA transcription or translation. Actually, I just realized we don't have as much time as I thought. So let's do until 15 instead. And so 4 minutes should be enough time to get through some of this. That way we'll have enough time for the Kahoot. We might still run a little bit long, sorry. So again, just as last time, be careful about the 5' and the 3' ends. So think about the template strand of DNA. Is it the one that is complementary to the mRNA, or is it the one that's nearly identical to the mRNA? And think about how the 5' and 3' ends change depending on what your answer is. And then your mRNA is going to be read in a certain direction as well. So it's also important to be careful of that when you are answering your question. Is the mRNA strand anti-parallel? So the mRNA strand is going to be anti-parallel to the strand that it's kind of being made off of. So if it's complementary, then it will be anti-parallel. Does that kind of help without giving away too much? Okay, great. All right. Hopefully everyone is kind of had a chance to try it out. Is anyone done? Would anyone like to volunteer for the mRNA sequence? Okay, great. Go for it, Isabel.
5' AUG, UGC, GGC, AGU 3'.
Yeah, exactly. Great. Okay, did anyone get to the amino acid sequence? Okay, go for it, Andrew.
Okay, so I got the MET thing, like MET, and then CYS, and then the GLY, and then SER.
Yeah, exactly. Great. Good job, both of you. And good job, everyone else, whether or not you got it correct. I think it's a really good question to attempt. And so here is the solution. So when you transcribe it, into mRNA, because you're looking at the template strand, it's going to be complementary, which, as the question that was asked earlier, means the strands will be anti-parallel. So you see 3' here, it'll be 5' here. And then mRNA is decoded in the 5' to 3' direction, so you can just read it like this very easily. All right, great job. So the last thing to cover we'll cover that really quick. But the last thing to talk about are DNA mutations, and I'll try to finish that in the next 4 minutes, but we will see. So I wanted to do a real life connection slide and wanted to talk about hair color because I keep on using this example, but it's not like I don't really give all of the biologically accurate details. And so I don't want people to be confused. And so I wanted to talk about this because it's a good example of how gene expression can actually affect something that we can physically see. And so with hair color is determined by melanin pigments, as we've discussed in the past, except melanin, these melanin pigments, they're not actually proteins. And so what affects the levels of melanin or the type of melanin that we have and what ultimately affects hair color is actually going to be different proteins that are involved in the melanin synthesis pathway, so the making of melanin. And we're not going to go into detail about what the pathway is, but basically you have two types of melanin, two main types of melanin. One is eumelanin, which codes for black or brown hair, and then the other is pheomelanin, which codes for red hair. And so depending on the type of the melanin you have and the amount of each, you're going to have a different hair color, and that's shown in this chart here. So for most of us, we probably have a very large amount of eumelanin and thus giving us black hair. People with red hair are going to have mostly pheomelanin and very little eumelanin and etc. for brown hair and blonde hair. You guys can read this on your own time. And so the type and the amount, which determines our hair color, is actually going to be determined by multiple genes. And so it's not just, one gene, one hair color gene. That's not an actual thing. And that's a good question. So yes, both eumelanin and pheomelanin are variations of melanin. So they're the two types of melanin pigments that you have. And so one of the genes that helps determine your hair color is the Mc1r gene, which encodes for a receptor called the Melanocortin-1 Receptor. You definitely don't have to memorize this, just throwing some names around. And so this is a protein. So it's expressed during the gene expression process. And this receptor is going to be involved in melanin synthesis and the production of melanin. And it actually determines the type of melanin you have. So it'll determine if you have eumelanin or if you have pheomelanin. And tomorrow when we talk about alleles, depending on which allele of the Mc1r gene you're going to have, that's going to affect which type of melanin you're going to produce. And so hopefully that kind of gives some more biological detail to what we've been using or very broadly as an example, and provide an idea of why it's important to have gene expression in the first place and kind of how it can affect individuals on a more visible level. And so the last thing to talk about, sorry, we're probably going to run long, but is our DNA mutations. And as mentioned at the beginning of class, these mutations are just changes in an organism's DNA sequence or in their genome. And there are two main broad categories that we're going to talk about, which are point mutations and frame shift mutations. And so these are different types of mutations. Point mutations are mutations where only one nucleotide is changed. And these are the most common because if you think about like the probability of one mutation or one nucleotide being mutated versus the probability of many nucleotides being mutated, I think it kind of makes sense that the probability is going to be lower if you have to mutate more nucleotides. And so we're going to talk about 3 subtypes of point mutations. And we'll also talk about frame shift mutation, which is when you have the addition or deletion of one or two nucleotides, or some multiple of, or some number of nucleotides that's not a multiple of 3. And you'll see why that is in a little bit. And so these mutations may or may not result in changes to your gene product because of the fact that we have so many codons that overlap and code for the same amino acid. And so that, if it does not have an effect, that would be a silent mutation, where your mutation in the DNA does not actually affect the amino acid sequence. And so it doesn't actually affect the protein. And an example of that would be like the diagram here. You have a T in your original DNA sequence, and then after a mutation, it gets converted into a C. And so if you have a change in your DNA sequence, that's going to affect your mRNA sequence, and in turn, possibly affect your amino acid sequence. So here in the original, you have CAU as your second codon, but here you have CAC as your second codon. But your amino acid sequence actually doesn't change, and both of these encode for HIS, right, HIS. And so the reason for this is because CAC and CAU, if you look back on the codon chart, are going to both encode for the same amino acid, and that's why it's called a silent mutation. Another point mutation called a missense mutation is where your amino acid actually does get changed and it gets substituted for by something else. And so two missense mutations are shown here. One is, or not here. So here you have the original DNA and mRNA. And so here you have a change from C to G, and then here you have a change from G to A. And so both of these are going to change the amino acid sequence of your amino acid, or it's going to change the amino acid in your amino acid sequence, and thus potentially affect your final protein product. And because we mentioned earlier that some proteins are going to have very different properties, some amino acids have very different properties from each other. So depending on if the new amino acid that gets substituted in has very similar or very different properties, that's going to affect like how severely your protein is impacted and changed. Nonsense mutation is the last example of point mutations that we'll talk about, or last type, and these result in a premature stop codon. And so rather than having, in this case, a 3 amino acid long polypeptide chain, you're only going to have 2, because this mutation from C to A is going to result in a stop codon, and it stops the translation process. And so your amino acid is going to be cut short. And so you're basically missing a really large portion of your protein. And that's usually going to result in a non-functional product. And then frame shift mutations, which is the last one we'll talk about, is when you have the addition or deletion of some number of nucleotides that's not 3. And the reason for this is because it shifts the reading frame. So your reading frame is your codons. And so for each codon, 3 nucleotides, that's going to be kind of 1 frame, and then et cetera, et cetera. And so if you have the addition or deletion of nucleotides that's not a multiple of 3, that's going to shift all of your codons starting from that mutation. So if you look at the example here, when you insert an extra A base here, then all of these amino acids are going to be changed, because rather than having CAU, you now have CAA, and then the U gets shifted to the next codon, which means it's also going to affect UAC, which becomes now UUA, and the C gets moved to the next codon. And so everything gets shifted, and it changes the rest of the amino acid sequence, and these usually have a very severe effect on the protein product as well. And so these are the 4 types of mutations that we're going to talk about, and we're now going to go do the Kahoot. So while I'm pulling that up, let me know if you guys have any questions about DNA, about DNA mutations or anything else we've talked about today. And for today, if you guys can, please stay behind, because we do still have 5 minutes, and the Kahoot today is relatively short. So let's see, question about what happens to the last A. So it depends on it depends on what your DNA sequence is, of course. So I think it would just not get read. I think you would either encounter a stop codon like much earlier than normal, or it would just not get encoded into the final protein because you just have one nucleotide, so it doesn't really affect much. Yeah. We'll double check on that too, though. That's a really good question. I haven't really thought about that. Let's see. Okay, if you can't play the Kahoot, that's okay. Please still do stay behind a little bit though, because you can see my screen and it's helpful for you guys to kind of see some of the questions and answers that we have. Start sharing again. There we go. Yeah, I'll give you guys like 30 seconds to join and then we'll get started. And so again, you guys are, fighting for a chance to not do today's homework, which, is nice to not do homework sometimes. Okay, take another 10 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Okay, all right, we'll get started. And I think you can still join in the middle of the day if you want to. Just for the sake of time, I won't stop for the scoreboard every time. So we'll see it like every 4 questions or so. Let's see the scoreboard. Okay. That's a third of the way through the questions. last 3 questions. All right, last question. Let's see the winners. There's more suspense today than usual. Oh, Alex again, good job. Okay, good job everyone. If you have any questions, let me know in the chat. Oh, yeah, that is strange. Oh, I think that's because of the, what's it called, the speed or something. But good, thank you for pointing that out. I guess we can make an exception for today, and both Alex and Isabel get out of homework free card. Ura, could you mark that down? Thank you. Okay, all right. So that's the end. Thank you guys for staying past a few minutes. Let me know if you have any questions. I'll stay behind a few minutes after class. Otherwise, I will see you guys tomorrow.